Great. Okay. I wanted to thank you very much for inviting me today. I haven't given a talk at Stanford in probably 10 years, but I'm assuming it still works the same. So you may have heard, I've been a research scientist at NASA Ames since I started as a postdoc there. I've gotten to the point that I just look around the room and say, before all of you were born. And then my follow-up is, it was a special kindergarten program. No, it wasn't. But anyway, my background really is in biology, particularly evolutionary biology, and particularly microbiology. And when I say microbiology, not bacteria as much, although we do a lot of work on bacteria in my lab now, but it's been more protozoa and algae, which have a lot more personality than bacteria. And so anyway, I got brought into Ames for their exobiology program, which as over the years has morphed into astrobiology. I was heavily involved in that, but I always loved teaching and having students. And so about maybe 20 years ago, there was a course in astrobiology at Stanford. In fact, it was the oldest course in the world on astrobiology. And I was asked to take it over. And at the time, it had started in AeroAstro. It had, at that time, was being run through HumBio. For some reason, I don't quite understand. 1 year it was co-listed with geology, which was great, but I hadn't initiated it. And then HumBio wanted to drop it and focus more on medical or whatever. So anyway, long and short is we need to find a new home, and it hasn't actually happened yet. But if you want it, I'm still game to do it because it was really fun, and I love really good students, and obviously they're great students at Stanford. And the one great thing about you, I mean, there are many great things about you, but I'm looking because Justin goes to Brown, so I have to be a little ecumenical here. But one of the great things is that you're local, and so you can come on over to my lab. I don't know if Advite's here yet. Oh, there. Oh my goodness. How could I have overlooked him? Advite spent what, a summer, I think, with me. I wish it had been 5 years. Still working on it. People who are really good, I put sort of like an ankle bracelet on so I can keep track of them for the rest of their lives, and hopefully bring them back. So anyway, we do have a lot of students, and then for about 9 years until COVID struck, I was running the Stanford Brown Stanford, depending on the year. 1 year with Princeton iGEM team, and that was really fun too. And again, really hope that we can start bringing some Stanford students back in the lab. So I'm not being totally altruistic coming over here today. I'm hoping that I get some of you really excited about some of the stuff that we do, and you might someday want to work with me, or encourage some of your friends. Or what I'm also really hoping is that we have a better contact with your student space initiative, which we did some work with. So I could talk for hours, but you don't want to hear me for hours. So what I am going to do is I'm going to talk about the whole sort of synbio and not really do heavily on astrobiology today, because I thought that that was what you wanted. I have sort of like a bonus talk at the end of some of what we were doing with BioLaunch, which was a fun project that we had with the AeroAstro, with the small spacecraft design group. I'm actually doing now more with AeroAstro than I did at the time, which was kind of funny. And so again, since it's sort of connected, and we've had students from this group in the distant past, I'm really thrilled if you would like to do some collaborations again. At the time, the issue was that they had a lot of people doing spacecraft design, which was great, but no payloads. And I don't do spacecraft design, but I've got a million ideas for payloads. So it was a really nice combination. And I know you're interested in biology, and I'm sure there are others as well. So again, delighted to help, and hope that this is just the start of a conversation. So without further ado, can we turn off the lights, and you will be on your own reconnaissance not to fall asleep, or if you fall asleep, please don't snore really loudly. Let's see, maybe we can get even a few more off here. Whoa. Okay, so this is serious, like no snoring. Everyone raise their right hand. Okay, so let's see if I can also remove some of these other people. Hi, Latanya, I'm so glad that you were able to join. Okay.
Thank you.
Okay. Great. And, I'm hoping that this will be helpful to your students and that we'll have a follow-up conversation there. And so for those of you who don't know, there are a couple of students from Howard University, I believe it is, who are listening in from Goddard because they wanted the same talk this week, and I didn't really want to give it twice in 1 week. And so they're joining in. It looks like Nakumbu, I'm pronouncing it wrong, is on there. Okay, that looks better up there. It looks awful here, but that doesn't matter what it looks like here. Okay.
Can we try view full screen?
View
Right corner?
right corner, sorry.
Yep. I saw full screen option.
Full screen. Awesome, thank you.
Well, you know what?
I wonder if I
I'm trying not to say close enough for government work. As a government employee.
Okay, I'm not sure, sorry.
Okay, I think we're pretty close here. So we've done a lot of work in my lab on a lot of different topics over the years. I probably am best known for work on life in extreme environments. But my PhD, and periodically we do some work on evolution of photosynthesis. We've done a lot of work on DNA damage in nature and balloon and scientific payloads. The ballooning was, of course, the project that was started at Stanford, but we also did some in Maine as well with Maine Space Grant. We still are active in origin of life, synthetic biology, obviously, which you're going to hear about today, which includes the biomaterials, biomining, paper-based biosensors, all sorts of stuff. And so, as you can see, the beginning part of this was astrobiology. It then sort of morphed into synthetic biology. NASA is a government agency, and so when your management tells you, you will do this, you do this. And what happened was the center director at the time, this was about 15 years ago, told me to start a program in synthetic biology and gave me a grad student. And so I said, yes, sir. And off I went. And to this day, I'm still representing NASA on a bunch of working groups and so on. So you probably don't know it. There are various interagency working groups on different topics. And there is one on synthetic biology that's been on for most of the time since about 2011. And I've been representing NASA. So every other week, at least, we get on the phone and the program managers at DARPA and NIH and NSF and NIST, and occasionally we get people like, from FDA or, all these, acronym agencies that float around, and we touch base and say, gee, what's going on in your agency? Or many of us work together on the executive order that came out on the bioeconomy last fall. And so we were touching base on that and how we respond to it and so on. So that's really been very interesting and a great privilege. So I did get a list of questions that people had submitted, and these were the ones that I could see from the list were astrobiology. So I thought I will save you the agony of not knowing whether I'm going to answer your questions. So I'll just answer them now at the beginning. So if you then want to leave very loudly, you can. No, I hope you don't. Is astrobiology something I could do in the future, even though my background's in EE? Absolutely. I had actually a Stanford student who his undergraduate, I think, was bioengineering, then got a master's in EE, and that was perfect for doing paper-based biosensors. So there are all sorts of skills, and if someone is clever about it or has an understanding of the higher level viewpoint, there's probably virtually no skill that someone in this room has that we couldn't use. So absolutely, EE will work too. And by the way, you probably know, organisms use electrical energy quite a bit. If you're assuming that you're still alive, you're generating electrical energy from whatever you ate last. To function with your nervous system and so on. It's not just the electric eel. So we worry about membrane potential. We worry, all those things that organisms have going in addition to using it as an enabling technology. How could students get involved in astrobiology? Well, one thing is I'm pretty good at finding students, but a lot of my colleagues are not as good. And there are various NASA intern programs. And what you need to do is go to the website and take a look. Some of them are paid and some of them are not. Obviously, I think the best ones are locally at Ames. But there are also many universities that now have programs in astrobiology. And quite honestly, there's a lot of research going on that with people who don't label themselves as astrobiologists, for whatever reason, but they're really doing astrobiology. But if you really want to go into a career in it, I would strongly urge you to have a home discipline. So, be really good at planetary science and then, reasonably good at about 5 other things. Hopefully I didn't say anything offensive. And, wow, that doesn't usually happen that early. So, be really good at biology, but also speak a little mechanical engineering and geology and so on. And particularly people who are undergraduates, but also graduate students. A lot of these fields I think of as languages. And if you have an opportunity to learn to speak them as a native, that's so much better than trying when you're 50, and you will get to that age eventually, then trying to learn these vocabulary words in geology, and it'll just sort of come out of your mouth in a more relaxed way. And so if you take a bunch of intro and maybe one upper level class in a bunch of things, that really will help set you up well, but do have a home discipline. The Stanford will make sure that you do anyway. Unless you want to actually major in astrobiology, and I'm not sure if Stanford would permit it now. I'm not sure whether Justin's planning to do that with me. I still have a faculty position at Brown, and that's why Justin's in my lab, and I have a lot of Brown students right now, and they can design their own major. And I have in the past, but that's when I had an actual faculty position at Stanford, did have a couple of students that I was the faculty advisor for who had gotten permission to design their own astrobiology major. So we may be able to do that with, with someone, combined with someone who is actually in the faculty now. When will we hear, what are the coolest research going on in astrobiology? Obviously, I think my work, so that's easy. Now, I mean, there's lots of really great stuff going on in origin of life, extrasolar planets. I mean, people didn't even realize that there was, there were actual planets there that's been suspected since the time of the Greeks, but no confirmation. Extrasolar chemistry, I think, is really exciting, and this is out of someone who never particularly cared for chemistry going through school. But when the field really got started about 30 years ago, 40 years ago, I had a colleague who used to come in and lecture when I was at Stanford, Lou Alamandola, and he said, when he started off, and you said that there was anything, even an atom in the interstellar medium, people laughed you out of the room. And now we know there's all this actually organic chemistry going on. So it's really exciting now seeing that this language of life is out there. So that's really cool. Always interesting work going on in molecular biology. There's a ton of great stuff going on, but this has also been enabled by the fact that we have many more missions going on right now so that we can get real data. When will we hear about aliens? So my dear friend Seth Shostak at the was it Seth or was it Jill? But anyway, one of the people at the SETI Institute used to say that the guesses were always about the time that you think you're going to retire when we're going to hear about aliens. So, do you have any water or something? I'm sorry, or anything. Okay. So the answer is I don't know. Are you interested in an alien who's going to talk to you? It's going to, take a class with you, be nice and furry and fuzzy, or are you interested in anything that we can identify as a life form? I don't know who answered that question, but people, I've been up on stage with a lot of these SETI people, and we sort of joke that, if you want intelligent life, you could score a home run and there could be some signal, and then, say it's a home run. If you're looking for microbial sort of life, any sort of life, which really the big difference is between non-living and living, then I think we have a much better chance. And as Seth says, you're looking for stupid life, I'm looking for intelligent life. But before we get too arrogant about it, people at the SETI Institute define intelligent life as a being who can make a radio transmitter. So with that definition, I am stupid life, and proud of it. I am part of a group that includes intelligent life, but I am personally stupid. So that's fine with me. But as I said, I really think that the big difference is between non-living and living. It is not between any living organism and something that's intelligent, because we know a ton of different steps that you can go through in evolution to get to intelligence, and intelligence is convergent. And that's a whole other separate lecture, but again, happy to talk about some other times. So that's about all I'm going to do with astrobiology for today. I'm sorry, but since I was invited to sit by, I'll do that. Say more than happy to come back and do all astrobiology sometime. So what we're going to do instead is focus on what it takes to get off planet Earth. So when we're on planet Earth, it's really easy to talk to all of you. I mean, it's, you know, this is trivial. The time delay coming out of the sound coming out of my mouth to your ears is insignificant. And even, you know, sort of more remarkable is the time delay between sound coming out of my mouth and getting to people in the East Coast who are listening in is also imperceptible. Our communication problems are not that great at this point on planet Earth. But as you leave planet Earth, and of course, you should all be able to recognize this Landsat image of the Bay Area, communication then starts to become much more difficult. And here's a great picture, of the Earth from ISS, and you can see the Earth is starting to recede into the distance. And the point here is, as you go further and further, here's a photograph from the surface of the moon. Again, these are actual photographs. You now have much greater distance. And so you need to have much more autonomy. And just, you know, because I work at NASA and I can do this, here's a picture from the surface of Mars. So can you find planet Earth in this picture? Yeah, I can do. Okay, I'll give everyone just a second here, and then I'll give you the answer. And so once again, as we move forward to ISS and then to the moon and then ultimately to Mars and potentially further in the way into the future, the Earth recedes into the distance. So the humans who are making this journey are going to have to be fairly self-reliant. But on the other hand, there's certain needs for humans. And it doesn't matter whether you're in Brooklyn, where this picture was taken, or you are on the moon or Mars. And some of these needs include things like transportation. You need to have life support. You need food, oxygen, medicine, waste, recycling, clothing, and so on. We don't think about oxygen here because it's a given, but it's not a given once you get into space. So that becomes important. In fact, gravity isn't even a given. We're going to need habitats, just like we do here. We need power, heat, light. We need radiation protection, and this becomes increasingly important as you go out into space. I mean, you think the UV's bad in Palo Alto in the summer. It's nothing compared to what you're reaching once you get outside the stratospheric ozone layer. And then on top of it, you're going to have ionizing radiation out there. And so radiation becomes a really big issue as you go forth from planet Earth. You need forms of communication. And just because this guy's waving a lot of cash, I remind you, as they say in The Right Stuff, no bucks, no buck Rogers. Thank you so much. Appreciate that. Okay, so we have these needs for humans, but we have different ways of dealing with it when we're off planet. So one of the big constraints off planet is what I'm going to call up mass, and that's tied into the up volume. And what we mean when we say up mass is the mass that is launched from the surface of the Earth against Earth's gravity well. So a lot of the propulsion, a lot of the mass of a spacecraft is involved in just getting up those first few minutes off the surface of the Earth, really counteracting gravity. Now, we call it mass, of course, instead of weight, because weight is mass times gravity, and you have a shifting gravity regime as you're going up. So you really want to stick with mass. So mass is extremely important to us. If you can reduce your up mass, that buys you a lot more capability for other things. So when I buy a piece of equipment for my lab, and it, you know, I don't even look into how much it weighs. We get a new freezer in, fine, it's 500 pounds, it's 600 pounds. You know, I don't care because I'm not personally picking it up. But if I need a refrigeration or freezer unit to fly in space, I want it as tiny and low mass as possible, because that's going to be intimately tied into the cost of the mission. And you also need something that you can store. You want it reliable and flexible. So you can't get to the moon or Mars and say, oh no, I forgot. Because as I always joke, you know, Amazon.com just does not have overnight delivery to the moon. And power, that's also something that obviously is going to be incredibly important. You have to have a source of power. And I've been saying increasingly in the last year that this is really the ultimate circular economy. You can't cheat it. You know, people say that they're doing a circular system on something like Biosphere 2, that, you know, fabulous system that was set up in Arizona outside of Tucson. Now that was a great model system, and they were hoping to have it in totally self-sustaining with different ecosystems and so on. It wasn't able to do that, and there was some, if you want to call it cheating, you know, having to leave and buy things and so on. You can't do that when you're off planet. You know, what you've got is what you've got, and so you better be able to count on it, and you better be able to reuse things. And so I work in an agency who thinks the answer to most things is to have an on-off switch. We're clearly an engineering agency. And so I've been trying to tell them for about 20 years now that there is another technology out there that would solve a lot of their problems. And so what I have is this technology in this black box, and it's programmable, and you figure, well, so are computers, you know, that's sort of basic in this day and age. And it's modular, also very handy because you want to have that flexibility off planet. But it's also self-replicating, which now puts that in a different category than your computer or your cell phone or any other piece of equipment. It's like, wait, self-replicating? And even better, it's self-repairing. And you know, you're now got these capabilities that are so far beyond the ones with the on-off switch. And by the way, it's got all sorts of other capabilities. And you know, you probably have figured out that what I've just sold you is life. And once you start to think of life as a technology, there are a lot of things that you can do to enable space missions. Actually, a lot of things you can enable, you know, an enhanced quality of life on Earth as well. So what is synthetic biology? Because we're now adding the synthetic biology to these basic capabilities of biology. And I like to use just a really basic definition: the design and construction of new biological functions and systems not found in nature. And this is one that the Brits came up with, and then it was in the White House National Bioeconomy Blueprint in 2012. But that's a lot of words, so, you know, think of it that way if you want. You're not laughing there. I told you not to snore, but you can laugh, okay? There's a big difference there. So if you start to think of life as a micro machine that does carbon chemistry and nanotech, you know, you're really off and running. So I usually at this point out that years ago, actually Drew Endy and I were asked to present to a committee of the NASA. Well, it's sort of the big overarching committee, the NAC, NASA Advisory Council, and we ended up being shoved off with the aeronautics guys. And the two of us, you know, did our best sort of, you know, wild idea things. And these guys, I thought we had blown their minds. And the chair of the committee then looks at me and says, so what's the big idea? It's like, I thought we gave you the big idea, but okay, you know, we can do Star Trek if you want to do Star Trek. And so the rest of this talk is sort of my shadow boxing with this guy, but obviously we came up with something a lot quicker, and they were very enthusiastic. So let's start with the fact that life actually is a material. And, you know, you can think of really obvious examples like wood and leather and cellulose. You know, there's probably very few of us, if any of us in this room, who are not wearing some kind of biomaterial, whether it's a watch band. I realize I'm very old fashioned wearing watches, or a shirt, or, you know, you probably have something on you that's made of cotton or leather or linen or something or another. And then you can add something like bioprinting. So you can now start to rearrange the conformation of these materials or the cells making them. And that's great. But when you start to look at these organisms that make these biomaterials, you start to look at things like silkworms and trees that make rubber and cotton plants and so on. These don't look like good ones to bring if you're worried about mass. So you want to put these things in another form factor. All right, I'm laughing because my students used to say I should stop lecturing and just tell stories. So when I was starting to sell this program, I was at NASA headquarters and there are mostly engineers in the room. And I dutifully said, but synthetic biology about every 15 minutes, because that's what I was there for. And finally, one guy just turned to me, sort of snidely and said, well, you think biology's so great. Do you think you ever could engineer biology to make rubber? I'm just looking at him, it's like, you do know that rubber literally grows on trees, right? That would sort of end the conversation. But every time I see this slide, I think of this guy. So yeah, I mean, biology is great. It does all these things, but we're not going to take rubber trees with us. What we are going to do is take something like Bacillus subtilis. So this is a bacterium that actually has the record for long duration flight. It was on the LDAT, the long duration exposure facility. And it was meant to be up there for about 6 months. It ended up being up there for nearly 6 years. For some reason, they couldn't go collect it. And you notice that some of these organisms have these really bright white spots. Those are spores. They can form endospores. And since they're found out in the soil in nature, you think about if you had to live out in the soil there, sometimes it's hot and dry, like all summer, and sometimes it's wet. You have to be able to deal with a very changing environment. And that's exactly why they make these spores. And it turns out these spores are really resistant to space environment, which is fantastic. And so I'm totally into Bacillus subtilis spores. And it's also a great platform for synthetic biology anyway. And yeah, I love my algae. I love my protozoa. These guys are not, heavy on personality. They don't bounce around or, they don't wave at you and they don't have cute cilia or anything, but they let you sleep at night when you do a flight experiment. So that's why I've sent them up on satellites. So what you do is you just program them to make these other things that are made by organisms that we can't take up. Now, if you think back to your basic, like high school or junior high ecology, you have this basic system on the Earth where we've got inputs of things like light and carbon dioxide and water and minerals that primary producers, things like plants and algae, take to make organic carbon. And that's eaten by the secondary producers, and they produce other things, including these biomaterials. And so we're saying, why not use a similar system off planet? It's worked really well here for over 4 billion years. Why, instead of worrying about all these mechanical systems, if we could have organisms that provide this interface between the raw materials off planet and the organic chemistry that we need for organisms, and then making biomaterials, why not? So this has been our vision, is to have some kinds of bioreactors with organisms that can do that interfacing. And then they produce things like sugars and so on, that other organisms that maybe are programmed to bind metals or make cotton or whatever are fed with these things. And then, as I mentioned, you could add bioprinting. And you mentioned that because it's not only cool, but I had a PhD student from Stanford who worked on that. And we have a patent out on it, in fact. So it's really a cool potential. You can do all sorts of really cool things with material science once you start dealing with organisms. But life also makes materials. For example, it can secrete chemicals. I mean, you think a lot of the pharmaceuticals that we use today are based on biologically produced ones, starting with aspirin, or if not older, and we're not even talking about the recreational drugs here. But it can also bind materials. Think about the fun. Well, let me get into this. I will show you in a second what I mean. And what this does is it gets into one of the first projects that I'm going to present. And this is a project that we've been working on for about 5 or 6 years now on habitats off planet. And so here's a great photograph from Apollo 11. I know the rest of you weren't alive then. I was. I stayed up practically all night. I was pretty young, but it was incredibly exciting. And there's a picture of, I think, Buzz Aldrin looking back at the lunar module, Lynn. Now, they were on the surface for, 18 hours or so? I mean, it wasn't that long. And if you're the first person to set foot on the moon, you could pretty much pretzel yourself into anything. This is obviously really cramped living quarters. In fact, I did a calculation once, and the volume inside the lunar module from Apollo 11 is about the same volume as a C block cell in Alcatraz if the ceiling was only 5 feet high. So we're not talking luxury quarters, but like I say, really exciting if you've got a short stay, you're the first person or the second person, or probably even the 20th person. But if you're really going to stay there from any period of time and with large groups and get anything done, you obviously need a much more extensive habitat. And so the traditional approach at NASA is to bring things with them so that you don't rely on making something off planet. But we think there's a better way because look at the mass for the a nominal Mars mission for habitat. So some of the new suggestions that are out there is having robotic construction, for example, of a regolith, that's the surface material. It doesn't have organics, so we don't call it soil, or building an ice house. And there have been some interesting proposals, but we think we have a better way. And for that, we turn to mushrooms. Well, actually, we don't turn to mushrooms. Mushrooms are just the fruiting body of filamentous fungi. And most fungi are actually these filaments underground. I'll show you a picture of the fungal, what they call mycelia, that I took on a microscope. So there's fungal mycelia. This should look shockingly familiar to Advite. And that is actually the body of the fungus. The mushroom, as I said, is just the fruiting body. And so what happens out in nature is these mycelia bind the soil particles, and it helps to access nutrients for plants. They do all sorts of cool things. But others have discovered that if you take, for example, sawdust or wood chips, and you put it in a mold and you seed it with fungal mycelia, they will then bind these, and you can get a material that is kind of like styrofoam. And if you do post-processing like baking, you can end up with harder things like bricks. And so we came up with this idea that this could be a fabulous way to make habitats off planet. And there are a whole lot of great potentials to this. I mean, it's low mass if you're taking advantage of water and minerals and so on off planet, inexpensive, totally green, totally sustainable. You could, in principle, compost the whole thing when you're done. It's totally green architecture. So people are starting to look into this on Earth. And this did start with the 2018 Stanford Brown iGEM team, I think. Yep, which Advite was part of. And so this then got picked up for a NASA Innovative Advanced Concepts phase 1. And we're now actually completing an incredibly competitive phase 2. And it's no secret, because I've been telling everyone our real goal now is to try to get some of these to the surface of the moon and prove that this really works. And so if you were an architect, you could go through all this. There are all sorts of advantages. You can make these at room temperature, and there's not much up mass, and there's heat loss, and capabilities are good, and they're not flammable, and blah, blah, blah. Anyways, you might imagine we have a couple of architects working with us, and Chris Maurer, who's an architect in Cleveland, made this little video that you can see. This is sort of our concept for the whole thing. So you can see a spacecraft on the surface dropping a folded inflatable that is going to then get some water externally. I think at one point there was sound on this, but it wasn't working this morning from my computer either, so don't worry about it. But the idea is then you could open this up now. We could inflate this pretty easily because the surface pressure is so low on both the moon and Mars. But our idea is to have algae in there, you give it a little water, and then they should be releasing oxygen as a waste product, and that should in itself inflate it and provide a really nice environment now for the fungal mycelia to grow and harden the shell, and you've got yourself a habitat. And so it's kind of cool because you don't have to worry about the landing location or anything. You can then build to wherever you are. So video. And we have, as you have to do for these sorts of things, and getting back to the person who asked about EE, we'll skip that. We have a collaborator who has a company that specifically works on inflatables for space. And so this is a time-lapse video of one that he made for us that you could then grow the mycelia in. Now you need to have something like an inflatable because the atmospheric pressure is so low on the moon, actually, and on Mars, that you don't get liquid water stable on the surface. So you need the pressure, and also when you go to Mars, you can't just throw stuff out in the surface because of planetary protection protocols, and that's a whole other story. We also are working with a group at McMaster University in Canada. They're not too far from Toronto, but they have a fabulous planetary simulator. And so we're able to test some of these samples with a simulated lunar or Martian environment, which is really nice. And there's Chris, oops, he's coming up on your screen. There's Chris on the right, who's the architect I was telling you about, and there's me looking very happy. This is one of the prototypes that he built with his architect students, and this is ungrown, and there it is a few weeks later with the surface having grown more. It takes just a couple of weeks to grow these things, but obviously there are things that we need to do to see if we can grow them faster, and what species you're going to use, and what sort of substrate and so on. So one of the things the iGEM students did is get at this question of what kind of substrate. It's fine if you want to use sawdust in the earth, or people say use something cheap like grass clippings from your lawnmower. It's like, newsflash, there are no grass clippings on the moon or Mars, and there's no sawdust at your local hardware store. So I pretty much emptied out my closet and gave the iGEMers all these sorts of things. And the answer is that fungal mycelia will eat literally anything. As people ask me, how expensive is this to grow them? It's like they eat garbage. They literally eat garbage. But what was really exciting is they also would grow on both Mars and lunar regolith simulants, just adding a little nutrients. So we're back on that in that biz right now, because think if you could get some of the regolith material in it and then use that as a binding material, sort of like a concrete. I mean, how amazing would that be, right? And so sorry, I was going to swap I have two movies of this wonderful stool the students made for me, which is still in my office, and Advice stars in one of them, but this is another Stanford student. And so you can see this little stool that they made in 2 weeks is totally suitable for the average Stanford senior to sit on. I've even stood on it myself, and it's been fine, and it's been so popular, and so many people oohed and awed over this little thing. But it's great because there's nothing like actually seeing something, picking up in your hands to realize that, hey, this stuff is real. For NIAC, we also have to do some earth-based applications. It's kind of fun because we've hooked up with a restaurant in the Basque region of Spain, Azurmendi, which is the top sustainability restaurant in the world. It's one of the top in general. And so we've designed some banquettes for them, which we're hoping to also build at Ames, and start testing new myco-based foods. We have when I say we, I think I signed some form that I am co-supervising a PhD student at the Basque Culinary Institute. So, I mean, I signed things, and anyway, but I think that's really cool that I have a student at the Basque Culinary Institute, and that's Alessandro. So she's working on that kind of stuff. So that's sort of the big materials thing. So let's get into a lot of other things that are going to go much faster. Life does chemistry. It does chemistry on the Earth. A lot of the things in the Sigma catalog were made by organisms. There's no reason not to use organisms to do chemistry beyond Earth. I mean, you think about whether it's citric acid or, acetic acid, there are a ton of things that organisms make. And, I rest my case here. Now, the other thing, as I mentioned earlier, is that organisms are also the source for most of the pharmaceuticals that we have, whether they're making them right now or they were the inspiration from traditional therapies, and then they're made transgenically or made, chemically now. At least they were at one point biological compounds. And so as we're now looking towards these Artemis missions to the moon and then ultimately beyond to Mars, we do need to be much more serious about medications. At this point, people can get back to the Earth really quickly. And obviously the astronauts go through all sorts of medical exams before they go. So illness is not as big an issue, but it's going to become a much, much bigger issue. Now, the problem is a lot of medications have a limited shelf life. Actually, part of the problem is that the FDA doesn't test them for more than, I think it's 3 or 4 years, because they figure, you don't need it. You go back to the pharmacy. But if you're thinking about a Mars mission that might be 2.5 years round trip, plus you've got to load in advance, and what if you miss a launch window, and what if all of a sudden you're dealing with a much longer time period. Plus you're being exposed to radiation, potentially cosmic bursts or solar bursts. And so you don't really know for sure. And then those are the stable ones. There are whole classes of drugs that are really unstable. For example, biologics that are made by organisms, particularly protein-based biologics. Think something like insulin or teriparatide. So it just so happens that those are the easiest for us to make in the lab. And so what we proposed was something called the astropharmacy, which actually is a legacy of the 2019 Brown-Stanford iGEM team. And that also got turned into a NIAC phase 1, and then we've just been awarded a phase 2. And as ultimately happens, we've done these great proof of concepts with iGEM, and then we bring in, a whole bunch of collaborators to fill in parts when we have a serious proposal at NASA headquarters. And so I've got a bunch of people working on, including pharmacist Phil Williams is one of the top schools of pharmacy in the world, University of Nottingham. And then various people, Mike Jewett's just in the process of moving to Stanford, but he's been working on us since he was at Northwestern, et cetera, et cetera, et cetera. And so this has been really cool. Basically, if you've worked in a bioengineering lab, it's fairly trivial to program a bacterium to make a protein. And so we're focusing on these protein-based drugs that are normally only stable for about 6 months. And, again, you have a long trip to Mars or whatever, you need a lot longer than 6 months. So what we've done is taken a couple of these drugs, and what we're doing is engineering Bacillus subtilis, back to Bacillus subtilis, to make them and secrete them with some tags, and then develop microfluidics-based systems where we then do potentially as few as 2 purification steps, and we're ready to go to give it to a human. So I don't give drugs to humans. That's not my biz, it's not my qualification. But we can certainly get to the point where we're checking for the activity and the purity, which is really cool. And I'm not quite sure why others hadn't really thought of this, but great. I mean, this is fun. As one of my former students said, we're the idea lab, and I love to be the idea lab. You can also use life for material extraction and separation. I mean, think about this. Even if you're eating a sandwich, you're not using metabolically every last atom in there. You're extracting what you need, and you're not using what you don't need. But it goes beyond just your sandwich. Think about metals. Now, we extract metals from the ground right now using heavy mining machinery. And as you can imagine, I'm going to say too massive, we can't do this. And then separation is not always very good. Again, usually massive facilities. This is Mountain Pass Mines outside of Las Vegas. It's one of the only ones that has a source of rare earth elements in the United States. This is a huge, huge operation. You couldn't take a fraction of that off planet. That would take multiple missions just to build the infrastructure. It doesn't make any sense. And, again, you've got these big chemical separation procedures and so on. Now think again back to your body. You use metals in a whole lot of ways. You may sort of vaguely remember from biochemistry, oh yeah, we got these cofactors and they bind this and they bind that. And, you need magnesium for chlorophyll, you need iron for hemoglobin and so on. Now focus on that instead of having it in the back of your mind, in the front of your mind. Could your blood cells say, today I don't feel like using iron, maybe I'll try cobalt, or, 95% iron's good enough, we'll use cadmium and the others. No. You don't get, in life, you don't get to pick and choose. We have unbelievable specificity in binding for our various biomolecules. We have no choice or we're dead. And this goes for something, I skipped over the chemistry, but polymer chemistry, a chemist who is in a lab and they're trying to make a copolymer. And so they need maybe 99 or 100 of one monomer and 200 of another. Okay, so it's 95 or 101 or whatever. That's not going to make any difference for what they're doing. If your DNA decides, oh yeah, 95% accuracy is good enough. No, you're dead. one base pair off, you can end up with sickle cell anemia or who knows what. So we have that kind of specificity built into life. And so if you can harness this power, think what amazing things you can do. And that includes, again, the biomining. So there are all these elements that we're going to need off planet. And this is just a list of some of the top ones. And there are a whole lot of elements that life normally uses, plus there are a whole lot that they use, including overlapping in some cases, or at least it's known, or you can make something that binds that. So again, we had a NASA Innovative Advanced Concepts, NIAC, on biomining project that started again with a Stanford Brown iGEM team in 2012. This was one of their projects. I'm going to actually mention all three of them. And what we did is we started with the biomining technology, but NASA wanted to see a real application. So we said, well, why not do an end-to-end system for recycling integrated circuits? So here's an integrated circuit. And yeah, you could take up a lot more, but if they're susceptible to radiation damage, they're all going to be bad. So it doesn't matter how many you can take up. But then you start to design, say, a peptide that can bind copper with 100% specificity. And so you grind up your integrated circuit, and you then, and Advite also worked on that. You grind up your integrated circuit, and you have a metal binding peptide, for example, binding copper. And then you take this and say you give it to a colleague who has a plasma printer, and they use that as the ink to print a new integrated circuit. You now have an end-to-end system. And so we've had a phase 1 on that. We haven't tried for a phase 2, mostly because we've gotten money from other places, but we need to get back to that. And so this is, again, I don't mean to keep picking on Advite, but not really picking on him. He had a fabulous summer with us. He, as I recall, came in one day and said, fungi can bind metals. I do it right. And he said, I was reading this book and they can bind metals. And I tell this story all the time, but not usually with Advite present. And you know what? He kept talking about this stuff, and it was, I'm sure, brilliant, but I wasn't listening to any of it after that. All I got was the fungi bind metals. Oh my God, we can combine that with the biomining project, and we have a filter. And so that's exactly what Advite did the rest of the summer. And I will blow his cover. He wasn't even 18 when he was working on this. We designed a filter for binding metals, and it is, we had a paper out of it, and this has now been at the US Patent Office. And hopefully it's very close to being granted a patent, because it's been there since, March of 2020, October 2020, whatever it was. October 2020. And we have had some inquiries, and in fact, we got an inquiry just 2 days ago on licensing it. So I think it's a really cool idea that we're moving forward on. And that also came out of an iGEM project, and actually then combining the two, and, heavy, again, Stanford involvement. But imagine how cool that is if you can just take the surface of a fungus and functionalize it so it becomes the substrate for mining, and then in the end, let's say you could compost it. It really doesn't matter. And it just, I really think it's revolutionary. So we've looked at all three of those first ones. We haven't looked at life for material production as much, but we have a big proposal, actually the Air Force right now, using organisms as scaffolds, not just for the biomining, but also to build for nanotechnology to actually, for example, there was another iGEM team in the Netherlands that made an E. coli, a bacterium that could bind silicon on its surface. And so you, in essence, had them making themselves into little glass beads. And they were doing this to make a lens, but I thought, ooh, how cool. So this has been in the back of my mind for a while. So we're heading to that, which ties into something like nanotechnology. But the nanotechnology project I'm going to show you instead, since the other one we're waiting to hear about, was also started by an iGEM team. Simon Vecchione was the captain of our 2013 team. He was actually he was a senior at Brown at the time. He then got a NASA fellowship to continue this work for his PhD at Columbia, and now is a postdoc at NYU and still working with us, which is fantastic. He is wonderful. But what he wanted to do is build a radio transmitter. And as I told you towards the beginning, I can't build a radio transmitter. If someone handed me a kit, that's not my thing. So we talked him off the ledge a little bit because I didn't think this was really feasible for 2 or 3 students in a summer to get some results. And I said, is there some part of it? So he had read about the potential for using DNA as a wire. Now think about a wire. Wire's a long, skinny thing. DNA is a long, skinny thing. Why not? And it turns out, and this is really funky, is if DNA, if you have a cytosine, instead of binding to a guanine, it binds to another cytosine, you get a mismatch, you get a gap. And what's really cool is that silver specifically will intercalate there into that gap. Who knew? And sort of also cool, if you have a thymine-thymine mismatch, you get mercury intercalating. So now you can see you not only have a way to build a wire, but you also have a way that you could potentially do some biomining with it as well. So again, he's had, I don't know how many papers out of this now over the last 8 years or so. He's kind enough to make me a co-author on a lot of them. He said, but it was your idea. It's like, no, it wasn't. He said, yeah, don't you remember talking to me? And it's like, I don't think so, but okay, that's very nice of you. Anyway, one of the things, we do go back and forth, and I'm very naive about it, and sometimes things that come out of my mouth make sense, and sometimes they don't, but I'm glad sometimes they do. But anyway, he's gotten very good at NMR, and it turns out that's one of his images, that you don't get a single silver atom, you get 3 in every mismatch. And if you think about it, now you have a tunable wire. So you can decide exactly how many silver atoms you want and where with atomic precision. So how cool is that? You can't do that with a copper wire. You've got a copper wire, or maybe you solder in some silver, who knows what. But with a chain of DNA, you can say, all right, we're going to do 3 mismatches, and then we're going to have a 2 base pair clamp, and then we're going to have 15, and then we're going to do this. And that's the kind of thing we've been playing with. For his PhD, you also start playing with making 3-dimensional objects, so DNA origami that then was metallicized. And I'm sure that someday this is going to be picked up as a really cool way to make things like circuits, because think about how cheap DNA is and how tiny it is. I really think there's something there. Now, mention electricity, that we're all generating electricity from what we've eaten before. And again, I've had students working on that in the lab from Stanford. We, of course, send digital information, which is completely massless, to a DNA synthesis company. They make us DNA. And there's no reason, if you're sending it to a company on the other side of campus or the other side of the bay, you can't send that information off planet. So it's really massless production. And that may not sound like a big deal to anyone in this room, but think about it. Up till now, throughout the entire history of life, there's always had to be a physical continuity. And now we've broken that link where you can send the information and then physically make the DNA at destination. So very cool. Why not create organisms that can survive in other sorts of conditions? So better adapted to surviving on the moon or whatever. And this was another project for the 2012 iGEMers, which they called the Hell Cell Project. And I use that name. I've used it up at the White House. Everyone loves the name. I still love the name. My current postdoc insists on calling them synthetic extremophiles, which is so toned down, but I love the Hell Cell. And he did get his degree at Stanford. Oh, well. Anyway, these students came to me with a bunch of genes that they found in the literature that should help confer desiccation resistance, radiation resistance, pH resistance, heat, and cold resistance. And particularly at the time in 2012, I thought, this is absurd. There is no way that 3 students are going to transform E. coli with all these genes and test them by the end of the summer. But I'm also a mother. Both my kids, as of 2 weeks ago, have PhDs. And by golly, I know that if you tell a kid that they can't do something, they will work twice as hard to prove you're wrong. And they did. And they proved I was wrong, and I was thrilled that they proved that I was wrong. And so this is, for example, some of the results you get with desiccation. So there's that blue bar is E. coli, which is kind of a wimp that grows in your gut. It's used to an unlimited buffet and constant temperature. So it's pretty much a wimp. So that's the control in terms of resistance to desiccation. But if you start to add things like the trehalose biosynthetic operon, you see an increase in resistance. Glycine betaine, which is a lot of organisms use for desiccation resistance, it just goes way up. And the manganese transporter, which we were working on for other reasons, oddly enough, also gave desiccation resistance. So, again, with very little time, very little work, we were able to shift the ability of E. coli to deal with desiccation, heat, and so on. And so this is something that I've had in my mind for a long time. And I now have a postdoc working on it, this health cell project. He's using other sorts of approaches with functional metagenomics, and we're really hoping to build that project up. We do, if you're interested in that kind of thing, we do have this in several proposals right now, and with a little luck, we'll have enough money to hire grad students and undergraduates on that. But you could build a cell from scratch. Why just alter one that you already have? And there are a lot of people, both in the origin of life community, which has been primarily funded by NASA for 50 or 60 years now, as well as now the synthetic biology community is working on building cells from scratch. In fact, Justin and I are going to Minneapolis next week for the Build a Cell and the SynCell meetings. Kate Adamala is the PI on a NSF big grant for the Build a Cell in the United States that I'm co-PI and so on. And we have these meetings about every 6 months. The SynCell is more of a professional meeting. But the reason I'm taking the time on this is if you're interested, she's now been running a virtual seminar series on Mondays. Well, for us, it's Mondays at 9 a.m. And they record all these, so you can always go hear the talk later. But every time I listen in, it just blows my socks off how much really amazing research is going on around the world. And a lot of times these are people I've never heard of, research I've never heard of. And so this is really cool that all this is going on. And so again, anyone is welcome to tune in and listen. It's I would say a large part students and postdocs who listen in, but there are people of all ages and levels and so on who do. And again, you can go to the Build a Cell website and you can find recordings of all these seminars. So I think that we are getting close to this building a cell. So I've probably gone a fairly long time, but I do want to finish by telling you that a lot of people management pat me on the head and say, well, yeah, these are crazy ideas, Lynn. Now it's time for the big boys to do this stuff. And so what we need to do to prove that this stuff works is to actually take it into space. And in, I think it was 2012, I got a call from our chief scientist, and he's on a Friday night. He said, the center director and I are in Germany, and we think that we're going to get an offer for a secondary payload on a German satellite Monday. What do you have? Preferably SynBio. Like, what? I mean, this is not how missions are supposed to work. But I spent all weekend glued to the sofa, and I thought, well, we do have a project that would be basic SynBio, ready to go in a way, in that we've thought about it. And that was the very first iGEM project that we did, the power cell, which the 2011 Brown Stanford iGEM team came up with. And so their idea was that this would be a biological, like a nerve center, or like a power plant, a biological power plant for a SynBio-enabled Mars colony. And so that's what we proposed. It turns out they'd called 2 other people, and then there were 3 secondary payloads, and then there were 2, and then there was 1, and that was us, the power cell and Eucropus. And what was cool about this satellite mission is they weren't doing any radiation, which I would have loved, but what they did do is look at different gravity regimes. So if you're not spinning, you're mimicking microgravity, which would be like the ISS. Then they spent about 6 months spinning to mimic lunar gravity, which is about a 1/6 Earth's gravity. And then they spent about 6 months mimicking Martian gravity. So it's about a 1/3 Earth's gravity. Now, to cut to the chase, The primary payload was using a simulated urine to grow tomatoes at the 3 gravity regimes. And for some reason, there was something wrong with their software, so they never got a communication. So in the end, we were the payload on Eucropus, which is named after their payload, the Euglena crops in space, which never worked. So we went back to this original idea of ours where we had a power cell, we had a photosynthetic organism as that interface with the raw materials on the planet, and then producing other things, feeding production cells. And in the end, what we had to do, and this was done heavily with a lot of actually Stanford graduates who had started with me on iGEM team and then spent a couple of years working in my lab to really get this going. So we had the power cell, but since I was given, or we chose to do a microfluidics card, so it looked like, if you're familiar with biolabs, a 96 well plate, except it was a 48 well plate. And so there are 4 rows of little wells with 12 wells each. And basically all we could do is a little fluid flow, temperature control, and each well had a little teeny tiny spectrophotometer built in. So you could measure optical density changes and color changes. So you can do a lot with that. You just have to be clever the way you design your experiments. So we did a transformation, and we also looked at promoter strength. And did it work? I won't spend a lot of time on this because, all the details, because I know it's getting late, but the power cell part worked. It actually was fairly gravity independent, which was great because then we can do the experiments in the Earth, which is a lot cheaper and quicker, but, good to know. Transformation, we did get some, we had to do it very differently. If anyone here is used to something like electroporation, you should see an engineer's face when you tell them you want a jolt of electricity through the works. It's like, no, that's a hard no, 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 no. So we ended up finding an Bacillus subtilis, I hate to use the word mutant, but a construct that had been deposited in the collection that could become competent for transformation by adding a particular sugar. And so that's where the fluid flow came in. So we made it competent with the sugar and then took care of it that way. And, I'm speaking to people in the room who are biologists. Never hesitate to call a culture collection. Culture collection, the curators of those are like, the scene in the first Harry Potter movie where he goes to Ollivander's and he gets his first wand. That's what a curator culture collection is like. They're like Ollivander. You say, I need this. And I said, I think I have something in the back that might be perfect. And that's exactly what happened. I mean, no one had ever heard of this construct, and it was perfect. It was just what we needed. Do not hesitate to write or call these people. That's why they're there, right? They just thrilled. The promoter was sort of iffy in that we'd expected a really big effect of gravity, and we saw no effect. So, I mean, just maybe there's something wrong with the promoters. Okay, so I've given you a ton of big ideas today that are really tied to space exploration. Obviously, synthetic biology can be used in aeronautics. People forget that the first A in NASA is aeronautics. We are the National Aeronautics and Space Administration. So clearly biofuels is something of great interest. The Japanese have run whole flights on biofuels. It could fit into green aviation and so on. Being a federal employee, I can't compete with private industry, but I think there's a lot there that we could be doing that's beyond the biofuels. And then, of course, there's the science and there's the astrobiology, which I mentioned a little bit at the beginning. So let me give you a few final thoughts here. I started off telling you there were all these constraints I have to deal with. Now, people on the Earth have other constraints they have to deal with, the environmental constraints, which are very different from ours. A big constraint of working on the Earth is geopolitical considerations. There are already probably existing infrastructure to solve the problem, and so you're now having to compete with them. You're having to worry maybe about investors. There are all these issues. I have other constraints that are completely different. But I feel like my constraints, once you sort of get a handle on them, can unleash creativity, because now I can think about personalized medicine. I can think about all sorts of things once I'm dealing with miniaturization and modularity and so on. And so I firmly believe that because I have these different constraints, it's forcing us and the space game to think of completely novel solutions that will ultimately also be brought back to Earth and really, really make a difference for those of us living on planet Earth. So that was what I mainly wanted to say to you, except Ad Astra, because that's to the stars, that's us. Thank you to, a cast of a gazillion. We are, as I said, still working with people in Aero Astro. These were the many, many iGEM projects that live forever on their wiki. And there were a whole bunch of questions that people sent me or sent here for me to answer. And I don't know, I think some of them I hope I have already answered. I'm happy to try to go through these quickly. Stability is obviously an issue. So one issue is to keep your genetic constructs in Bacillus subtilis spores. Integrative engineering so that you've actually put the genes inside the chromosomal material rather than plasma. It also increases stability. Circular habitation systems, obviously that's the kind of thing we're interested in. Mechanical devices being used for these studies. I'm not quite sure what you're getting at there. Foolish revelation, obviously, again, it's intellectual mother in me. I think all of these are really cool. It depends where we're getting good results or where I've written a proposal each week. Right now I'm on this whole scaffolding thing because I'm really anxious to see if the money comes through. I'm really, always up on the health cell because you can see that's not just for synthetic biology. It also is astrobiology application. And in fact, we're waiting to hear about funding for that from a program at NASA headquarters that has nothing to do with technology. It's called Habitable Worlds. And the way we tell them we're interested to see what the limits of life are, whether it's feasible for an organism to live on the surface of, Europa, Enceladus, whatever. Biggest challenges you're facing in your current research. Being shut down for 23 months in COVID, that was horrible, horrible, horrible. I lost a lot of equipment. A lot of it's the money. We don't have as much money as people think. I've heard people say we have 25% of the federal budget. In our dreams, we have 25% of 1%. Happy to take donations. It's a really cool agency to work in. So that's probably the biggest challenge that we're now building up. And I have a certain number of colleagues who just gave up and retired. And I still have too many ideas and too much enthusiasm to do that yet. And so I bit my tongue and we built back up and we got cool people in again. So, hopefully more of these will be funded and, maybe some of you will join my lab and get back in business. Fungal spores into space. I already talked about that. I don't know if the student from Jen Brophy's lab's here, but love to talk to you more offline. What can be done in space that can't be done here on Earth? I don't know yet. I'm assuming that's a scientific rather than a political question. Current state of mammalian cell research. I don't do mammalian cell research. But that's a great question. I know people who do, and happy to point you those directions. Now, we are, we're late. I'd rather take questions, but I did want to just remind you again that we had this fabulous ballooning program going with Aero Astro that includes the Space Initiative students. And, I really, really, really would love to get that going again. So I won't take any more time. Having spent time today making pretty slides, but the basic point is that if you get up to 100,000 feet, and I think our record was 120,000, but that may have been from Black Rock rather than, from the Bay Area, but it was the same group of students. And once you're that high, you've gotten through most of the stratospheric ozone layer, so you're now at about the atmospheric pressure of Mars. And you also are at about the same sort of levels of oxygen and radiation zone that we think we're on the early Earth. And so you're basically using it to both travel in time and travel in space. And that to me is why it's particularly cool. It is the fastest way to test things in space and in time. So it's the astrobiology and the synthetic biology in a 2.5 hour round trip. You can't beat that. And the NASA ballooning people keep saying, why aren't you applying to our programs? Because I can do it right here with no money, putting my payload in just as the students are letting go of the balloon rather than going through, X thousands of dollars and trying to fly out wherever you are. But that being said, NASA for years has run a ballooning program around the Antarctic. And so you think about if you're in the poles, you can stay up much longer. And we did try to get on one of those missions at one point. They mostly are doing big radiation payloads. But that would be obviously very cool if we could be up on one of those for a long period of time. I get really nervous when students say to me, but we could be up longer. I mean, we'll collect the balloon in Finland or something. It's like, my blood pressure always goes up when these balloons are up anyway. I show you one picture. This was just our first one. You can decide when these things burst. You can't necessarily decide where they're going to land. And in the Bay Area, speaking Spanish helps. Let's see, we had the mission control from Durand. But that was the end of the first flight. So you can see we need all sorts of skills, including the ability to climb a tree. We had one that was filmed by, I think it was the BBC or it was some British company, and the balloon ended up landing in a gated community in the payload in the East Bay. And we had to talk our way in from the guard at the front gate. And then, my nightmare scenario is it landed in someone's fancy pool and we'd have to knock on their gate and say, that thing that just fell from the sky, don't touch it. In the end, it ended up in the woods. Okay, and it was still beeping, so that's fine. This is now late at night. And there were rattlesnakes out there. And obviously I also don't want to kill off students with rattlesnakes. In fact, I don't like to kill off students at all. I try to avoid this whenever possible. And so we didn't go out that night. We went the next day, and this was a film crew, and their presenter then got on and filmed this thing because they were supposed to be in San Francisco the next day saying, well, this is emblematic of synthetic biology, that we're a long way from an actual success. It's like, no, wait a minute. We didn't launch till 2 p.m. because you didn't like the scenery of where we launched. This has not been a failure. I literally stood in front of the camera, which I've never done this before, and said, no, stop. This is not on us. This is, but again, you have to be ready for landing in all sorts of exotic places. And my kids always like me in the chase car because you never knew where you're going to end up. But it really is a lot of fun, and it is scientifically fantastic, and it's a wonderful learning experience. So again, I can't stress enough that if you people have an interest in having that going again, that would be so great. I would just love it. And I'm in. So that's what I have to say, which was really fast and probably like feeding from the fire hose, but now you know what we're all about and why I think, doing biology in space and, using space as sort of a frame of reference for astrobiology, synthetic biology, and so on is really cool. So thank you so much for inviting me. Again, happy to take questions. I actually live closer to Stanford than I do to Ames, so happy to come back, whatever. I know it's late at night. I don't know exactly what your rhythm is right now, whether you're getting ready for finals or whether you're sort of pre-final and not worrying and totally cool. I'm happy to answer questions or gab or whatever you want for a little while longer.
So
Yeah.
the problem with ceiling we're working on?
Yep.
What sort of genetic modifications do you reckon?
None at this point, actually. So we've been working on Ganoderma lucidum, which is a bracket fungus, and we've now starting to do some work on Aspergillus because it is much more genetically tractable.
Thank you.
You're quite welcome. Bye. Thanks for coming. Yeah.
I was like, and you guys were talking about you don't think of oxygen as being something you need because it's just so prevalent. And once you said organisms have to speak this, and then you think about is it possible to anticipate everything that an organism will need to survive until it send it out to space, and then a couple of nights you go, oh right, you forgot this thing. How do we know everything in advance?
So for an organism, you know, we grow them in these chambers, we've got a pretty good idea of everything they need. You know, we know who needs oxygen and who doesn't. Now, we may have to do experiments to see what level of oxygen we can bring them down to. I mean, there is still a report from, I think it's like 40 years ago, of growing an alga at 100% CO2. And so they made their own oxygen and were able to survive till they were kicking that in, which was pretty amazing. So that sort of thing you have to do an experiment to try. Radiation, you have to do you know, some of these things that we're not used to expecting, we do need to try, but you know, a lot of things are known that we're going to need a certain amount of something, and then it's just a matter of ballparking how much. Does that help you?
Yeah, sure.
For the biowires, have they been tested with electricity? Like,
Yeah.
do they connect?
Yeah.
That's so cool.
Yeah, I mean, that was what I was really hoping for that summer, because that would have just like, and so this gives me a chance to tell you that research is not always linear, and it's not always, you know, eureka moments. We ended up, that was one of those years that we had 4 different projects, and we probably should not have, and that's on me, but the student, I'm very bad at saying no when they're really great ideas. And so this was one of 4, and the other 3 were working, but you know, they weren't going to get Nobel Prizes, but they were working. This one was not working at all. They weren't creating, you know, any sort of wire forget conductivity, which was too difficult. And so there was Simon, and then Emily, who was also at Brown, who was an undergraduate in physics, and then a 3rd student. And so finally in August, so they were leaving in 3 weeks, I said, look, we need to think really hard about this. Do you want to keep going and risk not having anything to show for your project at the competition jamboree? Or do you just want to switch to another project? And the 3rd student said, I'll do whatever's best for the team. Emily said, I will do whatever Simon says. That's why they eventually were called Simon Emily, Emily Simon. Yeah. And Simon said, I'd rather have nothing to present. And I said, okay. And about a week later, all of a sudden, everything worked. And this is the only one that I'm still talking about now, 10 years later. And this is the one that's turned into all the publications and stuff. So sometimes, you know, you have to sort of go with your gut and hope that something works. And, you know, that's up to having a good advisor. And just, we were lucky. But if there hadn't been anything in another 2 weeks, I probably, with 1 week to go, would have had them at least had some marginal, you know, participation in some of the groups that would present. But it is just a really lucky. They look so, I mean, Simon looked so crestfallen. He said, please just give me another weeks, and it, you know, by golly, it worked brilliantly in the end. Not everything works right away. Yeah.
So thank you so much for the talk. It was really incredible to hear. And with the mycelial structures, especially, so on planet, I'm sure that definitely helps with like radiation protection, especially for long-term planning. Do you see that being used at all with like spacecrafts and in routes to Mars? And how might that look like?
Yes, because we've been starting to think about using that in aircraft. So, you know, you think about all that foam and insulation, or just even, you know, padding in an airplane or in a seat or whatever. There's no reason you shouldn't be able to use fungal mycelia for some of this. And again, the genius is that you can then switch it around. It's not permanent. But yeah, we've thought a little bit about it. We're definitely going that direction. Although oddly enough, my next 2 wild idea proposals, one of them is going to be on laundry. And that almost got funded last year. You want to know something horrific? We did calculation. If you have 6 people going round trip to Mars, so it's 2.5 years, and they change their clothes at the same rate they do on ISS, which is not very often. I mean, they change their underwear, I think, every day, and their socks every other day, but sweaters like every 6 months. Okay, 6 people, 2.5 years, you end up coming up with almost 2 megatons of laundry, 2 megatons. And yeah, that, talk about a lot of mass. And what they do right now is the cheap stuff they deorbit, which is a nice way of saying throw it overboard. And the expensive stuff like spacesuits they bring back, and it's like, that's absurd. And so there are a couple of really high tech, very cutting edge technologies that are starting to be developed for the Earth using, for example, liquid CO2, which we know we can make on Mars because we have with the Moxie. So that's what we've been proposing, is like, this is a problem you haven't even thought about. And God, this was awful. So apparently one reviewer said, well, there are lots of other ways to do it, and we have the secret pact anyway, and you know, and they send me this web link that's dead. And then someone else on the committee apparently was on an analog mission and one of these things in Hawaii or whatever, and said, well, I don't know why they have to do laundry at all. They should just deal with it. They're astronauts. Oh God. One of my administrators said, I'm sorry, that's not your job to explain to them why they have to do laundry. That was their mother's job. So apparently I have to be a little blunter when we resubmit. So I say, you know, I present this as great success stories, but you know, I've gotten my fair share of rejections too. We all have. And you get really pissed off for 24 hours or more, or some multiple of 24 hours. And then you know, calm down and you look at the reviews, and sometimes they're really stupid and sometimes they're good. But the ones that are really stupid, what they tell you is you were not clear. And so you don't necessarily have to change your project, but you obviously have to make it clear enough that even an idiot can understand that you have to do laundry. You can see I'm still pissed about this one. But we fully intend to succeed this year. Yeah.
Just a quick
Sure.
follow-up question on that. Is the laundry, would that be the like fabric itself is made out of mycelial material?
No, this is just laundry technologies. This is a total pandemic proposal. This does not use at all my background of biology. It is the first one. Did you know, for example, fun fact, that the average mass of a medium-sized pair of cotton drawers for men is 90 grams? I bet you didn't know that. And so you find that you're getting preliminary data, taking the kitchen scale and going up into the closet. But in fact, that was the same number that was online, so I felt better that my experiment matched the online data. Women's are a little less. Throw in a bra, you can get to about 80 grams, you know, and then you start multiplying times X number of people for 2.5 years, and the numbers start adding up. And you throw in the socks, and they, you know, anyway. Yeah.
For the astropharmacy.
I think you had a question.
It was the same one.
Oh, okay.
Sorry. For the astropharmacy, you mentioned, like, how does the microfluidic component work the filtration?
Okay, so what we were doing, actually, we were doing it with a lab at Stanford that helped us with microfluidics. And the idea was to just do a two-step, to have I don't know how much biology you've had, put on something like a his tag. So you add a couple of histidine residues and it will bind to a nickel column. And so you pack your microfluidics with the nickel column. And then the second was the sizing column. And you can get pretty pure there. Now, we have talked to people at Merck over the years, and apparently for drugs that are injected, you have to be up at something like 99% purity. It's incredibly high, as you might imagine. It would be a lot lower and a lot easier if it was an edible, but the problem with an edible, you can't have an edible protein because you're just going to digest it. So there are ways that people are working on to sort of split the difference, but you know, other people are going to have to do that technology. But we're assuming that this might have to be an injectable and therefore has to be unbelievably pure. Now, if you were looking carefully, you may have noticed in that picture that I used syringes. I had this crazy idea because when you start doing it with microfluidics, you normally have to have big pumps. And every time those two iGEMers, there's one from Stanford, one from Brown on the microfluidics, every time they said, and then we attach a pump there, I just pictured this big thing that was on the desk. It's like you just destroyed the whole mass, you know, issue. And so again, in my naive way, having only had one engineering class as an undergraduate, I thought, well, why not pack the microfluidics in a syringe and use like your thumb or your elbow as the pump? Because honestly, if you don't have an elbow or a thumb left on that mission, you've got a much bigger problem than whatever drug we're making. And so there's actually an undergraduate at Yale I met who then turned that into her senior thesis, starting to look up, ways to do that. And she's on the proposal that just got funded, and hoping we'll bring her in and get her in grad school. We're having a little difficulty because she's Eastern oriented, but we can introduce her to California. I grew up in the East Coast, so it can be done.
We have to do a class at 7:30.
Okay, are you trying to come in for a class? Oh, okay.
Never mind.
Okay.
Awesome. Thank you.
Anyone else have any questions? I know this has gone really late. And Ivana, Latanya, I don't know if anyone there had questions or if you want to just follow up on a private conversation. Yeah. Okay.
I'll leave it up to the students, to Ivana.
Okay,
if they,
great. I know it's really late out there.
yeah, it's 10:35, but that's okay. We don't care. This was wonderful. Thank you.
Thank you.
I know that the planetary protection protocols, and
Right.
also with the health cell, like how close are we just to having life organisms that could naturally spread on a foreign surface? Do we have a sense of that?
Again, it depends which foreign surface. As I mentioned, the issue is liquid water really isn't stable on the surface, but there could be liquid water, underneath, underneath the surface. I've pushed colleagues for liquid inclusions in the ice in a polar cap on Mars. And they say, because the geologists are very generous people, they want to give me a global ocean. It's like, I don't need a global ocean. If you are 2 microns big, what are you going to do with a global ocean? 100 microliters is a global ocean if you're that big. And I said, just give me 100 microliters of fluid. We can do that, I think there was a joke at some meme that we should have t-shirts, I just need 100 microliters, something like that, of water. But so I think you could find places like that. But yes, the planetary protection that we're talking about, it's 2 parts. One is we don't want to contaminate any planet that has any potential for life. And conversely, we don't want to bring life back here if there's any chance it could contaminate the Earth. And so there are international protocols in place, and the United States is a signatory to that. So we absolutely follow those. We can't just go contaminate. So the moon is basically clear because the assumption is that it is dead and always been dead. So we don't need to worry about planetary protection on the moon. We do need to worry about that in spades with Mars, because there's always a chance. And wearing my astrobiology hat, it would be an unbelievable scientific tragedy if there had been life on Mars and then we contaminated it with Earth life and we couldn't distinguish between the two, or worse, we killed it off, as we have this real habit of doing. So anyway, that's why there's planetary protection protocols. But that being said, and that's why we need these robotic missions as much possible, as soon as possible, because once humans are there, obviously they're going to take priority. So you have some problem, or there's a venting that hasn't gone right or something, you're going to release microbes. And that's, again, they're going to take priority over some astrobiologist saying, you can't let an astronaut die. They're going to take priority. So anyway, that's what we're having to deal with. Asteroids, the previous planetary protection officer said to me, just send me a note which one you contaminated. But obviously it's slightly more complex than that, because Ceres, which is the largest object in the asteroid belt, we think has ice on it. And once you're talking about water there, even if it's frozen, and potential for organic matter discoloring the surface of the ice, you start to think, ooh, what a great place for astrobiology. So I suspect that if Ceres isn't called out by name, it should be. But a lot of the asteroids are fine. So, again, Mercury, have fun. Titan, another matter. Europa, another matter. Enceladus, another matter.
What, pardon my ignorance, but what scientific value would the discovery of life on Mars have?
Oh my goodness. It would be data point, it would be N2. I mean, think about it. We only knew about one solar system up to actually, we knew about an extrasolar planet 50 years ago, this person. But the whole solar systems, we didn't start to really know about till the Kepler mission, 10 or 15 years ago, launched at the NASA Ames mission. And all of a sudden, we now have literally thousands of solar systems we know about. And so when we grew up, there were all these stories about, well, when you have a solar system, you have the inner rocky planets that are too hot, and then you have an Earth-like planet, and then you have the larger ones and stuff. Well, it turns out we're atypical. That a lot of them have these giant hot Jupiters that are close to their stars, and the migration patterns, and so we're not typical at all. Well, we have all these ideas about how life formed, and interestingly enough, it comes up pretty much with what we have. But what if it starts to turn out, what if we find 50 different life forms or 100, and there are a whole lot of different ways to make an organism? That would be the most incredible discovery ever. And I'm at the point that I would just take a second life form. Not even 100. I mean, it is so frustrating to have no other outgroup. And so that's why it's so exciting. And maybe there are multiple ways to make an organism, but maybe it turns out that some of our ideas that, you have lipids forming a bilayer, and you have proteins, and maybe you have nucleic acids that are based on bases. Maybe it turns out that is the only way to do it. And if you have two life forms that do that next to each other that evolved independently, whoa, that starts to make you think, well, of course, they have the same raw materials as an input because we're getting the same meteorites and so on. And if there are two independent life forms on two planets next to each other, it's going to have to be all over. I mean, what's the chance of two right next to each other if life isn't all over the place? And it's a lot easier to originate life than we thought. So, I mean, these are really profound questions, and that's why the potential for finding a second life form is just so unbelievable scientifically.
If N does equal 1, shouldn't we have like a backup plan for panspermia?
The thing is that if N is more than 1, we may know in my lifetime. But we will not know in any of our lifetimes if it's N equals 1, because there's no way we will have been able to do that kind of search. Now, if it turns out ultimately that we are the only life form, then that means excuse the expression, the universe is our oyster sort of thing, we're the lords and masters, and maybe we can dominate. We can also decide if we have ethical concerns for an inanimate universe. But it is an interesting second possibility that we really are all alone and we're special. And that would be incredible too. And it would also mean that a lot of our ideas in the origin of life are wrong. Because if they're right, we should be able to form life, not easily, but
If there's life on Mars identical to life here, but it wasn't fast, is that more indicative of two of the same evolving independently?
If it were exactly, then I would be highly suspicious that was our cousin.
Okay.
Yeah, that we descended. And if so, it's potential that we descended from Martian life. And I say that not just to tweak you, but because Mars is much smaller, so it cooled down much quicker. So it actually ended up in conditions to allow life to evolve much sooner than the Earth did. And we know there are meteorites that have come from Mars here. There's potential that we have a few on Mars that came from the Earth, but it's more likely to come in towards the sun's gravitation. So, yeah, maybe we're cousins. I really believe we're using the same base pairs if we're both using the same 20 amino acids, if we're using left-handed amino acids and right-handed sugars, all those things. There's no good reason why we should have those things. And so that would make me really suspicious. It's been so much fun. You're welcome.